Hello learners, in this video we're going to be going through all of the foundation bits you need for your Edexcel Biology Paper 2. So this is a nice low-key, low-stress revision, so you can sit back and watch this, you can make some notes if you want to, and then you can go to the website, look at the predicted paper, and we've set 3 questions and flashcards that we've got for you.
Plants carry out photosynthesis using light energy absorbed from chloroplasts in the plant cells, carbon dioxide that they take in from the atmosphere as it diffuses through the stomata into leaves, water that is absorbed by osmosis from the soil by root hair cells, and that goes through the process of photosynthesis to produce oxygen, which is released into the atmosphere as it diffuses out of the leaves via the stomata, and glucose, which is then transported by the phloem to all cells to be used by the plant. Photosynthesis is the chemical reaction that takes place in plant cells that uses light energy, carbon dioxide and water to make sugars. The symbol equation is carbon dioxide plus water, light energy over the arrow, oxygen plus glucose, which symbol is C6H12O6. It's an endothermic reaction because it takes in light energy. We need to know how plants use the glucose they produce from photosynthesis. A lot of it is used up in respiration to release energy for processes in the cells. Some can either be converted to starch or used to produce lipids for storage. Some is used to produce cellulose in the cell walls. Some is combined with nitrates from the soil to produce amino acids. These can then be used to make proteins through protein synthesis. We need to know the factors that can affect the rate of photosynthesis. As you increase light intensity, it increases the rate of photosynthesis. As you increase the carbon dioxide concentration, it increases the rate of photosynthesis. Photosynthesis is an enzyme-controlled reaction, so increasing the temperature increases the rate of photosynthesis up until the optimum, and then it decreases if it increases past that optimum temperature. The amount of chlorophyll in a leaf or in the chloroplasts in plant cells, if there's more chlorophyll, then more light energy can be absorbed, so the rate of photosynthesis can be increased. We need to be able to explain what's going on in rate of photosynthesis graphs with the different factors. So for light intensity and carbon dioxide, the graph looks the same. There's an increase and then a flat line. On the increase or the slope of the graph at A, whatever is on the x-axis, light intensity or carbon dioxide, is the limiting factor, because as it increases, the rate increases. When we get to the flat line at B, another factor is limiting the rate of photosynthesis, not what's on the x-axis. So for example, it could be temperature. The rate of photosynthesis reaction with temperature on the x-axis looks very similar and can be explained in the same way as any enzyme rate of reaction graph. Initially, there is a low increase in the rate of photosynthesis when temperatures are low because there is less kinetic energy of the particles. Then we get to the peak, which is the optimum temperature and the fastest rate of photosynthesis. And then the slope of the graph goes down because higher than the optimum temperature, the rate decreases due to enzymes that control photosynthesis denaturing. These factors could be changed in a lab when doing the photosynthesis practical, or you could need to interpret how these factors might affect plants growing outside. When measuring the rate of photosynthesis in the lab, we can change various independent variables based around the factors that can affect the rate of photosynthesis. We can change the light intensity by moving a light or a lamp closer or further away from our plant. We should use a heat shield in order to make sure that only light intensity is being measured and not temperature. Different colours of light can affect the rate of photosynthesis, as more red and more blue light are absorbed and green light is reflected by chloroplasts. So we can use different colour LED bulbs or coloured filters in front of a white bulb. We could use a water bath at different temperatures to create different temperature conditions for our plant. Or we could change the carbon dioxide concentration by using different masses of bicarbonate of soda or sodium hydrogen carbonate, which we would dissolve in the water the plant is in. This test tube, you see, come up this test tube and measure that in a certain amount of time to be able to calculate rate. You could also use a gas syringe or a measuring cylinder to collect and measure the volume of oxygen produced in a set amount of time. Let's look at an example method with light intensity being the independent variable. The control variables will need to be anything other than light intensity that could affect the rate of photosynthesis. So we need to keep the temperature the same, the volume of water the pondweed is in the same, the carbon dioxide concentration, so the mass of sodium hydrogen carbonate used, the same, and the colour of light should be the same. The type of pondweed used or the same pondweed should also be kept the same. I've placed a heat shield, which is just a beaker of water in between the lamp and the pondweed, so that the heat energy from the lamp is absorbed into that water and increases the temperature of that water, but the light energy passes through to our plant. This means that only light intensity is the factor that's changing, and the temperature should not change with our pondweed. You could also use an LED bulb, which does not produce as much heat. My independent variable then is my distance from the lamp. As we move the bulb closer to our pondweed, the light intensity will increase. So we start far away, we measure the amount of bubbles produced in a set amount of time, then we move the bulb closer, we can use a ruler to measure the distance, leave the pondweed to settle for a small amount of time, at least 5 or 10 minutes, so that the rate of photosynthesis has time to change, if it's going to change. Then you measure the number of bubbles in a minute or however long time you're going to do again. And then again, you would repeat by moving the lamp closer again, letting it settle, and then measuring the number of bubbles in a set time again. Just be clear in the exam what you are measuring, how you're going to measure it, especially having to remember to measure it in a set amount of time if you're going to calculate rate. Make sure you're clear about what the independent variable is, what are you changing in the experiment, and making sure that you understand that all the other control variables should be kept the same if they're going to affect the rate of photosynthesis. We need to know how living organisms are organised, so from the smallest part all the way up to the whole organism. Cells are the smallest living unit and the most basic building blocks of life. A group of similar cells working together to carry out a function is known as a tissue. A group of tissues working together to carry out a function is known as an organ. Then groups of organs come together to form organ systems. Multiple organ systems work together to form the whole organism. So an example of a tissue is a palisade tissue layer in a leaf, which carries out photosynthesis. An example of an organ is the heart, which pumps blood around the body. An example of an organised system of organs is the nervous system. And then a whole organism would be something like a whole plant, a whole insect, or obviously a human. And so we need to be able to understand that these go in size order as well. So cells are the smallest, then tissues, then organ systems, then the whole organism. We need to be able to describe the structure and function of some different plant cells and tissues. Root hair cells are one example of plant specialised cells. You can see here I've labelled all the structures we were labelling on our basic plant cell, but there's some things that are different. No chloroplasts, this elongated shape, for example. All of this is to help the root hair cell carry out its function, which is to absorb water and mineral ions from the soil. Root hair cells are especially adapted for their function because they have lots of mitochondria to provide the energy for active transport of mineral ions into the cell, and they have this unique shape which gives them a large surface area which increases the rate of osmosis of water into the cell. Xylem and phloem are two other types of specialised cell in plants that we need to know. They're highly specialised because they're very different from normal basic cells, and they transport substances around the plant. That's their function. Phloem transports sugars and amino acids, and the xylem transports water. you join all of those specialised xylem cells together, they form the xylem tissue. So this is formed of the lignified dead cells that form this hollow tube that transports the water and mineral ions up from the roots to the leaves. The cells have lignin in their cell walls and they have no end walls between the cells in order to allow this continuous flow. In phloem, the cells are living and they use energy to transport sugars from photosynthesis around the plant in a process called translocation. This flow is two-way because sometimes tissues above and below the plant need sugars, whereas xylem is only one-way flow. And this time the cells do have end walls, but they have holes in. The roots, stem and leaves form an organ system in plants which transport substances. The main method of transport here is transpiration, which is the flow of water from roots to the leaves and is known as the transpiration stream. The process of transpiration is where water vapour evaporates through the stomata. When stomata open so the plant can carry out gas exchange, water evaporates from the spongy mesophyll cells through the stomatal pores. So to open, the guard cells must become turgid, so they swell up and they push apart from each other, opening the gap between them, which is the stoma. When they are flaccid, when they don't have much water in them, then they come close together and that closes the pore. We talk about there being a continuous column of water, or the transpiration stream, which flows from the roots up the stem and to the leaves. This is because as water is lost from the leaves, it needs to be replaced in those cells in the leaf from the xylem. So this pulls water in from the xylem. A continuous column of water is drawn up from the roots due to the cohesion of water molecules. This means that they stick together or they're attracted to each other. This is because they can form hydrogen bonds between the molecules. Transpiration is the evaporation of water vapour through the stomata of leaves. Transpiration, or the rate of transpiration, can be measured using a potometer. So you can see the diagram set up below. This is just an example. Different potometers can look different, but we need to understand that we have a plant shoot, which will have leaves and stomata that the water can evaporate from, and that is connected all the way with an unbroken column of water from the bottom of the stem through a capillary tube, which has an air bubble in it. That's the only air bubble though. And we can move the air bubble to the start of the scale with the tap. And the air bubble is next to the scale, so we can measure the distance that the air bubble moves as water is sucked up through the plant shoot. And we are relating the fact that if it's sucking up more water from the plant shoot at the bottom, that is because more water is being lost at the leaves. You need to be able to explain how different factors can affect the rate of transpiration. Air movement or wind, if this is increased, it will increase the rate of evaporation of water, so therefore increase the transpiration rate. Light intensity, if this is increased, more stomata will open, so therefore this increases the transpiration rate. Temperature, if this increases, then the rate of evaporation is increased. Humidity, this is the only one that does the opposite, and transpiration will decrease as humidity increases because it decreases the rate of evaporation. The endocrine system is a system of glands that secrete hormones directly into the bloodstream. The blood carries the hormones to target cells around the body where it has an effect. We need to be able to compare this system to the nervous system and the way it works. The hormone system uses chemicals, not electrical impulses, to send information. The messages travel in the blood, not through nerves. The response is slower in terms of time for it to actually take an effect, but it can affect multiple organs all at once. We've got a labelled endocrine system here, and you need to be able to label all of these on a diagram and also to know what hormones each of these glands produce. The pituitary gland is found in the brain. It's also known as the master gland because it releases hormones that act on other glands, causing them to secrete more hormones. The adrenal glands are found on top of the kidneys and they secrete adrenaline. The pancreas secretes insulin and glucagon. The ovary secretes estrogen and progesterone in females. The testes secrete testosterone in males. And then we have the thyroid gland, which is in the neck, and that secretes thyroxine. The pituitary gland secretes two hormones that are also involved in the menstrual cycle. These are FSH and LH. During puberty, in humans, reproductive hormones are released, which cause the development of secondary sexual characteristics. This causes physical changes in the body that prepare the body for sexual reproduction. The testes release testosterone, which causes sperm production, the male gamete, to be produced. The pituitary gland in the brain releases LH and FSH. FSH, or follicle stimulating hormone, causes an egg to mature in the ovary, and LH, or luteinizing hormone, causes an egg to be released from the ovary. The ovary releases estrogen and progesterone, which build and maintain the uterus lining. These hormones from the ovaries and the pituitary gland control the menstrual cycle. We need to know that the menstrual cycle is a roughly 28-day cycle, and at day 14, roughly halfway through the cycle, an egg is released from the ovary, and this is known as ovulation. If that egg is fertilized, then it will implant into the wall of the uterus and the person will become pregnant. If not, and there is no sperm to fertilize the egg, then the uterus lining and the unfertilized egg cell will fall away from the inside of the uterus wall. This is known as menstruation or the period. Contraception is any method used to try and prevent pregnancy. It's a form of controlling fertility, which is the ability to have children. There are two types or groups of contraceptives: hormonal and non-hormonal or barrier methods. The hormonal examples include the pill, the patch, or the implant, and some IUDs. They contain progesterone and estrogen to inhibit FSH and LH, so no eggs mature or are released from the ovaries so that they can't be fertilized by a sperm cell. One example of non-hormonal barrier methods is the condom, or any other physical barrier that stops sperm reaching eggs. The benefit of condoms is that they also provide STI protection. Spermicidal products can kill or disable the sperm so that they cannot swim to the egg. An IUD is placed inside the uterus to prevent an embryo from implanting in the wall. They can be copper or plastic, or they can be hormonal as well. So an IUD could potentially be in either of these groups. And finally, surgical sterilisation is a permanent solution to cut the tubes that release gametes. So to cut the fallopian tubes so that eggs cannot leave the embryo and reach the uterus, and also to cut the tubes that attach to the testes so sperm cannot be released either. We need to be able to evaluate these methods. So obviously the benefits of condoms are that they provide the STI protection and nothing else does. For most non-hormonal methods, that they don't provide side effects, which is a good thing, and they're often very effective. There can be some disadvantages with the IUD. For example, it can cause pain, it can cause bleeding. There is a risk of infection because you're inserting a foreign object into the body, and they can also change your periods as well, making them heavier or more painful. But they are a long-term solution for contraception. Hormonal methods have quite a few negatives because they can obviously have side effects. They can affect periods, making them heavier or longer or more painful. They can also cause things like headaches and risk of things like blood clots. And again, there is no STI protection with these, but they can be long-term. So things like the implant or IUDs can be in place for years. So it's not something that you have to take every day, remember as a tablet or anything like that. So that can sometimes be a good solution. And they can obviously be reversible because when you stop taking them, your body should go back to normal. Homeostasis is the regulation of conditions to maintain an optimum internal environment in response to either internal or external change. There are three main things that homeostasis works to control in humans: body temperature, which should be around 37 degrees in humans, blood glucose concentration, and water levels. Maintaining these is important so that enzymes and cells function properly in the body. Homeostasis is controlled by the nervous system and the hormonal or endocrine system through the same cyclical processes. So it starts with receptors. They are found in organs that detect the changes in the internal or the external environment. We call those changes the stimulus. Then they send information either via hormones or nervous impulses. They send this information to coordinators, which process the information. This could be the brain or spinal cord in the nervous system, or for example, the pancreas in the hormonal system. Then the coordinator sends out hormones or nervous impulses to effectors. Effectors are organs which respond to the stimulus, and these are always either glands in the hormonal system or muscles in the nervous system. And then the body responds to whatever the change was or the stimulus was. Controlling blood glucose. Blood glucose levels are monitored and controlled by the pancreas. When blood glucose increases after a meal is digested, the pancreas releases the hormone insulin. This is an example of what we call negative feedback because the change that happens in the body is reversed through this process. So it starts with receptors in the pancreas detecting the increase in blood glucose levels. The pancreas is also the coordinator because it releases the hormone insulin in response to this. The insulin is the hormone that then travels in the bloodstream. Insulin binds to the liver, which is the effector, and it makes them absorb glucose from the blood into their cells. The response then is that blood glucose levels decrease because excess glucose in liver and muscle cells is then converted to glycogen for storage. Diabetes is caused when the blood glucose levels are not being controlled properly. It can cause high blood glucose levels. There are two types. Type 1 diabetes is when there's not enough insulin or no insulin produced by the pancreas. The treatment is insulin injections to help lower blood glucose. Type 2 diabetes is when insulin receptors on target cells like the liver and muscles do not respond to insulin being made by the pancreas anymore. Treatment is a low carbohydrate diet and exercise. Sometimes, if this is unsuccessful, glucose lowering medication can be taken, but insulin is not an appropriate treatment. Obesity is a risk factor for type 2 diabetes. It can be correlated with BMI or waist to hip ratios. This means that we can show a causal link between a BMI or waist to hip ratios that indicate obesity, and as these get higher above that threshold, that suggests that we can show an increased risk for type 2 diabetes occurring. Respiration is not breathing. It's an exothermic chemical reaction that occurs inside every cell to release energy from sugars. Aerobic respiration happens in the mitochondria in every cell. It uses oxygen to break down glucose and release energy. The symbol equation is O2 plus C6H12O6, and then it produces carbon dioxide and water. We need to know the uses of the energy released from respiration in organisms. It is needed to carry out chemical reactions to build new molecules like proteins, carbohydrates, and lipids. It's needed for active transport to move substances against their concentration gradient. It's needed for movement, specifically muscle contraction, and also for keeping warm or maintaining certain body temperature. A reminder that any cell that carries out a lot of these processes, so active transport, muscle contraction in muscle cells, or building new molecules, they will have lots of mitochondria as an adaptation to make sure they can carry out more respiration to release more energy for these processes. Anaerobic respiration in animal cells takes place when there's little or no oxygen. It still breaks down glucose to release energy, but a smaller amount of energy is released. It happens in the cytoplasm of cells. So I've put in a box all of the facts we need to know about aerobic so we can start to compare it to anaerobic. So it happens in a different place. Anaerobic does not require oxygen. The products are different and the amount of energy released is different. In anaerobic respiration, glucose does not break down completely, but breaks down into lactic acid. Lactic acid is toxic if it builds up in cells, so it's removed by the blood to the liver. Anaerobic respiration usually occurs in muscle cells during high-intensity exercise, when there isn't enough oxygen getting to them. All exchange surfaces will have adaptations to maximise the rate of diffusion. So exchange surfaces are places in organisms where majority of diffusion happens, and these are why they have this adaptation. They will all have thin walls, a large surface area, and good blood or air supply. Thin walls, such as having one cell thick walls in the small intestine in the villi, helps to reduce the diffusion distance and therefore speed up diffusion. A large surface area, obviously we know, increasing the surface area increases the rate of diffusion. And a good blood or air supply is ordered to maintain a steep concentration gradient. So if constantly substances that are diffusing through are being moved along to create a low concentration gradient behind them, that means that diffusion keeps occurring in the right direction that we want it to happen. There are three specific exchange surface examples that we need to be able to explain and describe how they have these adaptations and how it makes them good at doing exchange. So for example, the alveoli in the lungs in humans, this is where gases diffuse between the lungs and the blood capillaries. The villi in the small intestine, this is where small molecules, so amino acids, glucose, fatty acids and glycerol, which are the products of digestion, diffuse from the small intestine into the blood in capillaries. And finally, leaves in plants. This is where gases will be diffusing in and out of the stomata and into the spongy mesophyll tissue layers, where they will be exchanged with cells that are carrying out respiration and photosynthesis. Surface area to volume ratio is where we divide the surface area of an organism by its volume. And we've gone with cubes here to give you an example, but the idea is that the larger the organism becomes, the smaller their surface area to volume ratio becomes. If you look at my cubes and if we think about diffusion, the distance between the centre of each cube and then to the outside or to the external environment is much greater in the larger organism with the larger length of size of the cube. So compared to its volume, the distance between its volume and all of its outer edges is longer. This means diffusion from the very inside of that organism to the very outside of that organism would be very slow because the distance is very long. So we can't just rely on substances diffusing from the outside into all of the cells in the body if we are a larger organism. Single-celled organisms like this example I've got here do not need an exchange surface like the ones we've just looked at, because they have a very large surface area to volume ratio, because it's a short distance between all of their internal sections to their external surface membrane. So it's really quick for substances to diffuse from outside of the cell to the inside of the cell, and therefore they don't need to have any adaptations to speed up that rate of diffusion. However, the larger an organism gets, the smaller its surface area to volume ratio becomes. So it needs these specially adapted exchange surfaces like alveoli, like villi, and then a transport system like our circulatory system in order to increase the rate of diffusion of substances into and out of every cell in the body, so that it can happen fast enough for processes like respiration and photosynthesis to occur. Blood is a tissue which transports substances around the body and plays a role in the immune system. The majority of blood is made up of blood plasma, which is the liquid that blood cells are suspended in. It's mostly water, but it contains all the dissolved substances that are being transported around in the blood. Things like glucose and other things like antibodies, for example, and other dissolved substances from the digestive system, like the amino acids and things that need to travel to and from your cells. The rest of the blood is made up of 3 different types of blood cells that are suspended in this liquid. They're red blood cells, white blood cells, and platelets. Platelets are involved in clotting open wounds to try and prevent infection or to prevent excess bleeding. Red blood cells are specialised cells because they have no nucleus. They contain lots of the hemoglobin protein, which is the red coloured protein that binds oxygen. They have a biconcave shape, which gives them a large surface area to increase diffusion of gases into and out of the cells. And their job is to transport oxygen to and from cells. There are 2 types of white blood cells: phagocytes, these engulf pathogens, and lymphocytes. These produce antibodies or antitoxins. Both of these defend the body from pathogens and prevent you from getting sick, so they play a part in the immune system. We need to know the 3 different types of blood vessels and how their structure relates to their function. Arteries transport blood away from the heart at high pressure. This is how I remember that A for arteries and A for away from the heart. They have a narrow lumen to maintain high blood pressure. They have a thick elastic layer so they can stretch and recoil to maintain high pressure. So the maintaining high pressure here is part of their function, and this is how it relates to that bit of structure. So if you're describing and explaining the structure and function of arteries, you need to talk about saying the narrow lumen is a structure. What does it do? How does it help it carry out its function? It helps to maintain high pressure. Lastly, it has a thick muscle layer to withstand high pressure. You need to use these words, otherwise you won't get marks. Veins transport blood to the heart at low pressure. They have a wider lumen to transport a greater volume of blood. They have a thinner muscle and elastic wall layer because their blood is being transported at lower pressure. So we do not need the same features we see in arteries. Veins have valves. I remember this with the V for vein and the V for valves. This is to prevent the backflow of blood due to gravity. Capillaries transport blood close to cells so that substances can be exchanged. They have a very narrow lumen to slow blood flow down to 1 blood cell at a time. This increases the time for diffusion to occur. They have thin walls that are only 1 cell thick to decrease the diffusion distance. And ultimately they join small arteries and veins together, so once exchange has happened, the blood flows into veins and then can return back to the heart. You need to be able to use all of this information to be able to compare the structure of the different blood vessels as well as their functions. We need to be able to label and describe the structure of the heart. First of all, with any heart diagram, always write on the right and left, the right way round, so it is the opposite to how your hands are on the page. Then we won't get mixed up with our rights and our lefts. The two top chambers are the atrium, and the two bottom chambers are ventricles. Think about it as A comes before V in the alphabet, so A is at the top and V is at the bottom. The blood vessel that brings deoxygenated blood from the body to the heart is the vena cava. It is a vein. Once blood has entered the right atrium, it's going to flow down into the right ventricle, and the contraction of the right ventricle will push the blood up and out of the heart through the pulmonary artery. It is an artery because it's taking blood away from the heart, and it is taking blood to the lungs to be oxygenated. When blood returns to the heart from the lungs, it returns in the pulmonary vein. So because it has been to the lungs, it is now oxygenated blood. This is the only vein in the body that carries oxygenated blood. The blood then moves through the left atrium, which contracts and pushes it into the left ventricle, which will contract and push it up and out of the aorta. This is the biggest artery in the body, and it takes blood at the highest pressure because it has to send it to the whole of the rest of the body. The left ventricle has a thicker muscle wall. This is so that it can contract with greater force, so that blood is forced out through the aorta at higher pressure, because it has to go a further distance to get around the whole body than when it leaves the pulmonary artery and just goes to the lungs. The human circulatory system is known as a double system or a double pump system because the blood flows through the heart twice, once to the lungs and once to the rest of the body. So the right side sends blood from the body to the lungs, and the left side sends blood from the lungs to the rest of the body. The heart rate is controlled by a group of cells known as the pacemaker cells that are found in the right atrium. If these cells stop working, they can cause arrhythmia, which is an irregular heartbeat, and an artificial pacemaker can then be fitted to control and regulate the heartbeat. The right-hand side of the heart pumps blood to the lungs to be oxygenated and to remove carbon dioxide. You can see a diagram of the lungs here. We should know the trachea, or the windpipe, branches out into two bronchi, and then the alveoli at the end of the tiny branches, which are known as bronchioles, and we have two lungs, one on either side. Here we've got a diagram of one alveolus from a bunch of alveoli and the capillary that's next door. You can see both of the walls are only one cell thick, as we mentioned, as this is an adaptation. We have deoxygenated blood coming from the heart on the left-hand side. When it reaches the alveolus, then diffusion of carbon dioxide from the blood into the alveoli will happen, and then oxygen will diffuse from the alveoli into the blood. This is where it will bind to the red blood cells, which will carry it and transport it back to the heart. Once the oxygenated blood will arrive back at the heart, then the left-hand side of the heart will pump this to the rest of the body. Investigating respiration rates. This can be done by measuring the carbon dioxide produced. So we can use yeast as our organism, and we can measure the volume of carbon dioxide produced and collect it in a gas syringe. We can use a water bar to control or change the temperature. And if we want to measure how the yeast carry out anaerobic respiration, we can add an oil layer to prevent any oxygen from getting to the yeast. In order to measure the rate, we would calculate the volume of gas collected and divide it by the time we collected it in. We can carry out a control for a comparison where we have either no yeast or dead yeast. You can boil yeast to a high temperature in order to kill it. We've got a water bath here to control temperature, but you could also look at something like the substrate type or the concentration. Instead of looking at microorganisms, we can also use something called a respirometer to measure the rate of respiration in living organisms that are small, like insects. In these examples, we'll look at how much oxygen they actually use up instead of how much carbon dioxide they produce, but you can do it the other way as well. So this can be done with small organisms like insects, or it can be done with germinating seeds. So in the bottom of the flask, separate from the organism so they're not touching, we could put a solution of, for example, potassium hydroxide, or it could be sodium hydroxide, in order to absorb any carbon dioxide that's released through respiration. This means that as the organism respires, it uses up the oxygen in the flask and it lowers the pressure in the flask because the volume of gas in there is reduced. So the liquid in the fluid tube, which is known as the manometer, moves towards the flask, which means that the liquid on the other side of the U-bend of the tube, next to the scale, will move down. So this time we're measuring the distance that the liquid has moved, or the volume, if you know the diameter of the tube, and then divide that by the time it took for that liquid to move that much distance. That will give you the rate of respiration. The control, you would normally put in an organism that is dead, if it's dead seeds, but for insects, we don't want to kill insects, so you would take something that's the same mass, but is not going to do any respiration, so for example, glass beads. This isn't changing the independent variable, it's just looking at their normal rate of respiration, but you could do something like changing the temperature to see if it changes the effect. You could also automatically measure the carbon dioxide or the oxygen rate using a sensor and a data logger. We need to be able to explain the body's response to exercise. During exercise, the body needs more energy from respiration. This is to help muscles contract. During exercise, the breathing rate and the volume of breathing increase in order to increase the concentration of oxygen in the blood. The heart rate also increases in order to pump more blood faster to muscle cells to deliver more oxygen and glucose. When talking about this, we need to use the word more a lot because your body always breathes and your heart always beats and you are always transporting oxygen and glucose around the body and your body does always need some energy. But when we are doing exercise, we need more energy because we're doing more muscle contraction, so we need to increase our breathing rate to have more oxygen and we need to increase our heart rate to transport more blood faster to the cells so they can have more oxygen and glucose in order to carry out more respiration. In muscle cells as well, we have a store of glycogen, which is broken down to release the glucose for respiration. Cardiac output is the total volume of blood pumped out of the left ventricle per minute. It can be calculated by multiplying the stroke volume by the heart rate. The stroke volume is the volume in centimetres cubed of blood leaving the ventricle, and the heart rate is calculated in beats per minute. If either of these increases, then the cardiac output, which is measured in centimetres cubed per minute, also increases. During exercise, the cardiac output increases because the heart rate increases and the ventricles contract with more force, so the stroke volume also increases. we need to know the organisation of an ecosystem, feeding relationships in an ecosystem can be shown in food chains. Food chains always start with a producer. These are always photosynthetic organisms, as these are the source of all biomass on Earth, because they can use light energy to synthesize molecules like glucose. The arrows in a food chain or food web show the transfer of biomass, and by biomass we mean living tissue that is made up of the molecules carbohydrates, lipids. Primary consumers are the first organisms that eat another organism in the food chain. They can often be herbivores, which means that they only eat plants, and they will be prey species because they will be consumed by other animals. The secondary consumer is the second organism to eat another organism in the food chain. They are predators because they catch and consume other organisms, but they can also be prey to organisms higher up the food chain. Tertiary consumers, and then we also have quaternary consumers as well. These are predators and they're often carnivores, so they only eat animals. The top predator in the food chain will be the apex predator, and they will have no predators that eat them. Competition. All organisms will compete with other organisms for resources in their environment. So for animals, they compete with each other for food, space or territory, and mates. And plants compete with each other for light, space, water, and mineral ions. Competition can either be between members of the same species, we call this intraspecific, or between members of different species. This is known as interspecific. Competition leads to adaptations that allow organisms to outcompete others in their habitat in order to survive. We're going to look at adaptations in more detail, but examples of things like storing food that squirrels do to make sure others can't have it and make sure they have a store for themselves, long roots of plants in the desert so they can get more water from other plants, and then things like horns or claws, adaptations for fighting that allow them to fight for other mates or other things like food. Abiotic factors are non-living factors that can affect the number of species in populations in an ecosystem. They can also lead to species having specific adaptations to survive in extreme habitats. Light intensity is an abiotic factor. It obviously affects photosynthesis and can cause sunburn in animals. Temperature affects all organisms because it affects enzymes. It also has an effect on the rate of transpiration. Wind intensity or direction, again, affects transpiration, could potentially affect flight, and it can cause exposure issues in animals. Moisture levels or water availability, all organisms need water in order to survive. Soil pH and mineral ion concentration. Again, pH can affect enzymes of organisms living in soil, and plants need mineral ions in order to survive. The concentration of oxygen is an abiotic factor in water for aquatic animals. And the concentration of carbon dioxide is obviously a factor, but only for plants because it can affect the rate of photosynthesis. This is mostly not an issue unless these plants are in some kind of greenhouse or container. Biotic factors are caused by living organisms that can affect the size of populations in an ecosystem. New predators. Prey species may not be prepared or able to adapt to avoid predation. New pathogens. Organisms will have no resistance to the disease if they've not been infected with it before. Amount of food. Limited food due to a reduction in population size can increase competition and reduce survival. Equally, when there's lots of food, population sizes can increase. Competition. Competition with other species for resources could lead to one outcompeting the other until there are not enough individuals of that species left to breed. This means that they could go extinct. An example I've got here is the red squirrel and the grey squirrel. There are very few red squirrels left because the grey squirrel is able to outcompete them in most habitats. We need to be able to explain how biotic factors can affect population size. An example with disease. What would happen if a new pathogen reduced the number of birds in this community? It would increase the numbers of snails and worms, as they now had fewer predators. But the number of lizards and snakes could decrease, as the eagle now has to eat more lizards and snakes to make up for the lack of birds, as they are one of their food sources. So when we look at food chains like this and talk about the impact of a biotic factor on a community, we have to look both above and below in the food chain. A food example, we need to know about predator and prey population sizes, how they're linked, and how they often cycle over time in stable ecosystems. There are certain rules that these predator-prey cycles always follow. As prey numbers increase, the predator numbers will increase because there's more food, so more of them are able to survive and reproduce, but there will be a slight delay. If there are too many predators, or as the population size of the predators increases, they might eat too many prey, and so then the predator population size will decrease because they now have a lack of food. When the predator numbers are low or have decreased, fewer predators means that the prey numbers can then increase because more of them can survive to reproduce. If you were sketching these graphs, remember there can never be more predators than there are prey. And also the predator graph should always be lower and shifted to the right. In an ecosystem, there are individual organisms, populations, which are a group of individuals of the same species, communities, which are all the populations of all the different species in an area, and the habitat, which is the non-living parts of the environment. An ecosystem is the interactions between the living and non-living parts of an area, so all of the communities in a habitat. Interdependence describes how species in a community depend on each other. They can depend on each other for shelter, pollination, seed dispersal, and food. If one species is removed, it can affect the size of other populations in the community. Stable ecosystems have balanced environmental conditions, and the species numbers in the populations are relatively constant. Stable ecosystems take time to develop, and so examples are often hundreds of years old, like large coral reefs or ancient woodland. Parasitism and mutualism. Parasites live in or on other organisms called hosts. Parasite takes nutrients from the host and it receives nothing in return. So this means there's no benefit to the host organism of having the parasite, and often it can be detrimental to the host organism. An example is fleas on cats and also on dogs and other animals. Fleas feed on the blood of the cat. They also live on the cat, which provides it a habitat as they are attached to their hairs, and they can jump large distances, which allow them to jump from one organism to another. Many parasites also lay large amounts of eggs to try and increase their chances of reproduction, just to make sure that their offspring have a chance of some of them surviving and finding a new host. Parasites often have special adaptations to take as much as possible from the host, but without killing it. Another example is a tapeworm. A mutualistic relationship is where both species benefit. An example is bees on flowers. Bees get nectar from flowers, which is their food, and the flowers get pollinated, so they get help with their sexual reproduction. Lichens are another example of mutualism. They are actually two organisms, an algae and a fungus, and these actually live sort of together in this symbiotic relationship. The algae photosynthesize and therefore produce sugars which they share with the fungus, so they provide it with food, and the fungi provides shelter for the algae from harsh climate. It's not possible to count all the organisms in a habitat, so sampling is used to estimate population size in an area. We normally use quadrats. These are a metal wire frame and there'll be a set size, so for example 1 metre squared, and you would count the number of organisms of each species inside the frame. Random sampling methods are used to estimate the number of a species in a certain area. This is also known as the abundance. As you can see in the diagram, we would set up 2 tape measures in a way of gridding the area, and then we would use a random number generator to select coordinates for the x and y axes in order to say where to place the quadrat. This ensures that the quadrats are placed truly randomly and therefore removes bias. Also, we should place a large number of quadrats. This is to improve our reliability of our experiment. Systematic sampling with a transect is used to see how a changing abiotic factor affects the number or distribution of a species, so where a species is found. So, as you can see from the diagram, we've laid a transect going away from the base of a tree, and we've done it twice. So you would lay a transect along a changing environment, so a tape measure, for example, along a changing environment, and then place your quadrats at regular intervals. That means they should be the same distance apart. So here we've gone for 0, 5, and 10 metres. So every 5 metres along my transect. You should repeat the whole transect in a different position. So as you can see here, we've got 2 of them. The change in the environment should be the same. So as we said, moving away from the tree and starting from the base of the same tree. But there should be no overlap of your quadrats, so you're not sampling the same area. For both, you would calculate a mean across your quadrats. For random sampling, you could then multiply it by the area of the field that you are measuring to estimate the total population size. For the transect, you would take the mean from each of the quadrats from the same distance along the transect to show the mean number of each species at each distance. Deforestation occurs to clear land for building and farming. Chopping down trees reduces biodiversity because habitat and food sources for animals are removed, and the trees and plants are killed when they are cut down. Another disadvantage of deforestation is that it reduces the carbon dioxide that's removed from the atmosphere through photosynthesis, as there are fewer trees. This contributes to global warming. The burning of wood or leaving wood to decay that's been chopped down also releases carbon dioxide. And removal of trees increases soil erosion and flooding. When we say that land is cleared for building or farming, in terms of farming, we mean to grow crops for biofuels, or to create flat land to farm cattle, or to grow rice. The farming of cattle and rice both produce methane, which is a greenhouse gas that also contributes to global warming. There has been a massive population increase in humans because we have better medical facilities and better access to food, so people are living longer. This means because there are more people, we need more space to live and also to grow food. And we also have increased living standards, so we want more space and more food. This creates pollution from mining, building, energy production, farming, and that all damages natural habitats on land and in water. And because there's more people and we have more stuff, we are producing more waste. All of this pollution reduces biodiversity because it kills plants and animals. Land pollution, which comes from filling up landfill with waste, but also toxic chemicals in the soil, which can come from things like pesticides and herbicides being sprayed onto the land. Water pollution comes from sewage or fertilisers from farmland, and both of these create a low oxygen environment if they run into water, which is done through eutrophication, and it causes the death of animals like fish in the water. Or it could be toxic chemicals from mining or from nuclear power stations. Air pollution is caused by smoke and gases that have come from burning fossil fuels. So the gases include carbon dioxide, but also sulfur dioxide. The smoke and actually breathing in the particulates can harm lungs of humans and other organisms living around towns and cities. And the gases, including the sulfur dioxide, cause acid rain, which damage trees, plants, and cause acidification of lakes and ponds. The greenhouse gases, including carbon dioxide produced, also contribute to global warming. We need to know how humans can reduce biodiversity, so have a negative impact on the environment. The first is eutrophication. This is caused by the runoff or leaching of fertilizers or other chemicals used like manure or sewage from outflows from water treatment plants. All of this increases the nutrient content of the water. Algae, or plants that live in the water, then use these nutrients to grow, and they grow rapidly, causing what we call an algal bloom on the surface of the water. This blocks light, so plants lower down in the pond can't photosynthesize anymore, so they die. And then decomposers start to decay this dead plant material, and in doing so, they respire and use up the oxygen in the water. When there is not enough oxygen left in the water, this will cause other larger organisms that depend on the oxygen in the water for respiration, such as fish, frogs, and other organisms, to die. This therefore reduces the biodiversity of the pond. A second example is fish farming. Fish can be farmed in large tanks or nets in lakes or in the ocean. The farming process produces a lot of waste. The animals themselves produce waste, but chemicals are also used, things like antibiotics, drugs, treatments for various pathogens, but they can also get parasites. All of this will then leach out through the net or be kind of disposed of around the area and harm marine life nearby. If the fish are fish like salmon, which live off other organisms, then wild fish populations are often depleted because they're caught and used to feed these farmed fish. They will want to feed them a high amount of food in order for them to grow quickly and to grow larger. Sometimes fish can escape and then compete with indigenous populations, especially if they're not native to that area. And also predators of the fish try and obviously get in because they want this source of food, and they can get trapped in nets and injured. This links to our third one, which is introducing non-indigenous species. So introducing organisms to an area that they are not native to, as in they didn't originate there and they didn't grow there or are found there naturally, means that they may start to outcompete native populations for things like food, shelter, and other resources. Because they can affect food chains and therefore ecosystems, they can lead to local extinctions of either other predators or prey species. Scientists and conservationists have put programs into place to try and reduce human impacts on biodiversity and help to maintain it. Breeding programs for endangered species to try and increase their numbers. This often happens in captivity in places like zoos. Protection and regeneration of rare habitats to try and make sure that they are no longer damaged anymore and potentially help them to recover. Reintroduction of hedges and wildlife corridors around farmland. On farms, normally only one crop or one species of plant is grown in large areas, so allowing these extra plants and trees and everything around the edges of the field can give homes and habitats and food sources to lots of other animals. Recycling, reusing and reducing waste to try and avoid landfill and reduce how much actual waste goes to landfill. Reducing deforestation and the carbon dioxide emissions by governments. This is normally done through legal arrangements and getting them to sign agreements to try and reduce their impact on the environment. The carbon cycle. All materials in ecosystems are recycled to provide the raw materials for new organisms to grow. The carbon cycle returns carbon from carbohydrates, lipids, and proteins in organisms to the atmosphere as carbon dioxide, so that plants can use it for photosynthesis to synthesize new molecules like glucose. Decomposers break down dead organisms and return nutrients to the soil for plants to take up through their roots. Photosynthesis is the only process to remove carbon dioxide from the atmosphere. Decomposers, bacteria and fungi release carbon dioxide as well when they respire during decay or decomposition. Fossil fuels and the production of fossil fuels like coal and oil takes millions of years to form, and this is trapped carbon from dead organisms that hasn't decayed. Wood burning and burning of fossil fuels for transport and in factories in order to produce energy, these are all human activities, and these are what put carbon dioxide into the atmosphere, but these are processes that humans have caused that have caused the carbon cycle to become unbalanced. The water cycle is important as it provides fresh water on land for plants and animals before it returns to the sea. The first stage is evaporation, where water evaporates from large bodies like the ocean due to heat energy from the sun. Then it condenses, so it cools down to form clouds. Water also evaporates from leaves through transpiration. Precipitation falls from clouds either as rain, sleet, or snow. And then the water percolates through rocks into the ground and runs off of rivers and ponds and other things like farmland back into the ocean. In the water cycle, water is continuously evaporated and precipitated. Precipitation usually provides fresh water, but in very dry countries, possible drinking water is in short supply. There are different ways of turning seawater, which is often in large supply around the world, into fresh drinking water. The first one is known as thermal desalination. This is where seawater is distilled. So we heat up the seawater, the water evaporates, and then the water vapour is cooled down and condensed, and then this is collected as fresh water. This is expensive as it requires a large amount of energy to heat the seawater. It also contributes to global warming as it uses fossil fuels. Another method is reverse osmosis. In osmosis, water moves from a dilute solution to a more concentrated solution, so from where there is more water to where there is slightly less water. In reverse osmosis, pressure is used to force water molecules in the opposite direction, from where there is a lot of water molecules to where there is less water molecules, or from a concentrated solution to a dilute solution through a semipermeable membrane. So the water molecules move through and collect on the opposite side as fresh water, and the salt particles stay behind. Nitrogen gas is converted to nitrate compounds by bacteria in the root nodules of plants or in the soil. Nitrogen can also be fixed by lightning and a chemical reaction called the Haber process. Nitrifying bacteria then convert ammonia to nitrates in the soil. These can be taken up by plant roots, and they will use this to make amino acids and proteins. Animals eat plants, and this is how the amino acids and proteins pass into animals. The bodies of dead organisms, including plants, animals, and waste products from animals, all contain proteins. Decomposers break this down to return nitrogen to the soil. This is in the form of ammonia, which nitrifying bacteria can convert back to nitrates for more plants to take up, and so the cycle continues. In anaerobic conditions, for example when the soil becomes waterlogged, denitrifying bacteria break down nitrates and can return them to the atmosphere as nitrogen gas. This happens obviously only in these certain conditions, but it's a bad thing because it can decrease soil fertility. Farmers can improve soil fertility or increase the amount of nitrogen by using different methods. One is to use crop rotation. This is where they cycle or rotate the crops grown on different fields, and they will use ones that have root nodules and nitrogen-fixing bacteria to put nitrogen into the soil, and then the next year they will rotate and have a normal food crop which will then remove the nitrogen from that soil to grow. Peas, beans and clover are all examples of nitrogen-fixing crops. Natural fertilisers can also be used. They're often made from waste or plant material that will decompose and slowly release nutrients into the soil. Synthetic chemical fertilisers can be used, like ammonium nitrate, which is made from the Haber process. These are expensive and cause pollution and can reduce biodiversity.
Ouch! This is why in some videos I explain scratches.